Qian, for the very kind introduction. Let me share my screen, and yeah, I'm really happy to be invited and give the presentation today. Of course, haven't left Boston Children's for a long time. I only came to Philly since last October to start my lab, so it's about 3.5 months so far. So yeah, majority of the things that I'm going to talk about here today are going to be the work that I did at Boston Children's during my postdoc, and I'll touch a little bit on our new directions that I will take more in my independent lab. So again, I'm a new faculty here at Penn and CHOP, and we have this new center for BRIDGE, which stands for Brain Research Initiative for Development, Genetics, and Engineering, which really summarizes what I will be doing in my own lab. But today we are going to focus on single cell spatial omics analysis on the human cerebral cortex development. So, of course, for today's seminar, not every one of us come from a neuroscience background. But if you're asked to picture what the human brain looks like, this image is what comes in your mind. So the bulk part of what you're looking at is indeed the human cerebral cortex, because it is the biggest structure in the human brain, and it is the entire outer thing that you're looking at right now. And also, it is characterized by this memorable complexity in its structure, both in terms of its surface folding as well as the internal layers. And when comparing between different mammalian species, you can see that the cortex has gone disproportionate evolutionary expansion in size as well as in complexity. So this is where you find the biggest difference between different mammalian species, and many would believe that it is because of the advancement of the cortex that makes us so unique. But of course, the basic functional principle of development of the cerebral cortex is shared between human and other mammals. So majority of the insights we can obtain is being derived from rodent models, most notably mouse and rats, and they have proven really invaluable. But there are times where we run into limitations in terms of the fidelity that animal models can be used to recapitulate human developmental and disease-specific context. For instance, a gene that has been extensively studied by the Walsh Lab at Boston Children's, ASPM, is known to cause very strong microcephaly malformation in the human patient when it's mutated. But when the same mutation is introduced in the mouse, the severity is much less pronounced. So telling us that there could be some opportunities, some advantages if we can study human cortex development in its own terms. And one of the key differences that separates between mouse and human cortex is the human cortex has underwent this very dramatic expansion in the surface area, which leads to that the areas of the cerebral cortex can be divided into different functional regions that cannot be mapped one-to-one with a mouse, and they have divergence both in developmental origins as a developmental process as well as the function. But it is well known that the human cerebral cortex can be divided with these areas that contain different neuronal subtypes as well as exhibit different morphology for the layer organization. For instance, the visual cortex located near the back of the brain has a very thick layer 4, while the primary motor cortex, which is located near the center, has a very thin layer 4. And if you look at these schematics, you can see that different neuronal subtypes are found within them. So this has been characterized even 100 years ago using these morphological methods to tell the cells apart in terms of their layer and their shape. But it has not been well understood how these differences emerge during prenatal development, because these morphological features are not very apparent. But the process of understanding different cortical area specification, or the process called arealization, is important because we know that neurodevelopmental disorders often impact specific cortical areas instead of everything together equally. So I'm giving a very dramatic example here. Say a gene called SEP85L, when it's mutated, it will cause this very dramatic smooth cortex malformation specific to the posterior part, but with the anterior part almost completely spared. You can see the lack of the gerification here. And this happens that's not only in one or two cases occasionally, but there are full cohorts of patients that exhibit the same specificity and the same carrying different types of variants in the same gene. So understanding the process of cortical arealization could really build the foundation for a more specific and targeted way of understanding disease. So I've spoke a lot about the uniqueness of the human cortex, and of course now we are in this new age, and here we are in the data science webinar, right? So we the most powerful tool at our disposal these days to directly study human brain tissue is to do high throughput profiling of single cell gene expression, where we can represent each single cell as a dot and analyze its expression of hundreds to thousands of genes to compress everything down into a two-dimensional space, group similar cells together with each other, and identify cell types such as this. But I myself am a developmental neurobiologist, and often when I see these type of plots with very fancy mathematics of comparing hyperdimension data to 2D projection and doing a lot of statistic analysis, I always question, how can I be sure that these are a real biology instead of computational or analytical artifact? So what we can be sure is that I'm a believer of seeing is believing. And when we can analyze the gene expression of cells in its nature context, where they form this tissue architecture, and we see the beauty that nature has created, I see that this got to be the truth. This cannot be an analytical artifact. So that is why I am particularly excited about using spatial omics to analyze the developing human cerebral cortex, especially with true single-cell resolution enabled by imaging-based spatial transcriptomics method, such as MERFISH that I employed. So we set our goal to build a extensive developmental atlas for the human cerebral cortex using frozen tissues obtained from the NIH Neurobiobank. And we wanted to cover different cortical areas as well as several different developmental time points during the second and third trimester. When we perform the MERFISH experiments, MERFISH works by detecting small transcript dots into a two-dimensional space by basically microscopy imaging. And it is key that we need to assign each transcript dot into the cell that they belong to so that it will enable single-cell analysis. But given the high cell density found in the human cerebral cortex during the embryonic stage, it is very difficult to tell each cell apart. That's why we collaborated with Dr. Carson Stringer to train under the CellPulse 2.0 framework, a custom deep learning model that is based on our MERFISH experiments to allow us to automatically segment millions of cells within our MERFISH data. And this is where we turned this challenge of high cell density into an opportunity, because the MERFISH measures all the cells within the tissue slice, which could be, say, 1 centimeter squared of area, and it can contain up to 500,000 single cells, giving us a incredible cost efficiency for both our time and money when we run these MERFISH experiments. So together, we, in collaboration with Dr. Mingyao Li's lab at UPenn, we analyzed a total of 18 billion single cells with MERFISH, really making it by far the largest data of its kind in the field. And yeah, we did all sorts of bioinformatics analysis to extract the biological insights from these massive data sets. So first of all, when we cluster the cells only based on their gene expression information, just like how you would do for a single-cell RNA-seq, and then we project back into the location of the cells, we can visualize all the beautiful tissue architecture that is found within the developing cerebral cortex as illustrated here. So again, these clustering itself is only based on gene expression, but not on the space. But we always see these cell types organized into very clear lamination. And we can focus on the cortical plate where the neurons are. This is a developmental structure that later becomes the gray matter of the cerebral cortex, where you have the so-called six-layer structures. But that six-layer structure has been defined based on the morphology that I kind of showed you earlier. During the mid-gestation developments that we are looking at around like gestation week 20 or 22, the cortical plate is just all these neurons that are densely packed next to each other without any morphological differences between the layers. It is really only until the gestation of week 34 that you start to see some earlier hints of the cells being spaced apart, and then some of the layer-specific morphologies start to appear. So it has always been questionable in terms of when is actually the distinction between these cortical layers established. But now with the power of Murphy's, we can see that the six layers of the cortex is defined as early as gestation week 22 by specific subtypes of molecularly defined neurons, excitatory neurons. And this is 3 months before the emergence of the architectural layers. So basically, we are able to uncover these developmental features that really precedes the morphological observation. And another interesting developmental intrigue in terms of how cortical layers happen is that this is a process that works by the neurons that they are not born locally on the top in the cortical plate. They are born down below in the progenitor zone, and they go through a process called radial migration, where newborn neurons always climb up a rope from these radial fibers and get themselves from the bottom to the top, and they will always go surpass existing layers of neurons to sit on the top. So it's like building a brick wall where you take a new brick and you stack it on top of it so that it gets higher and higher. That's why the layer structure of the cerebral cortex is built in the so-called inside-out order, where the deep layer, layer 6 and 5, are built earlier, and then you follow by layers 4, 3, and 2. Layer 1 is a bit of an exception, so we don't have to discuss that right now. So of course this principle has been well established based on the birth dating, but now with the Murphy's, we can also see exactly in the human fetal cortex development at what time is each layer being built, as we compare as the cortical layer could progress over time. So the structural complexity of the developing cortex expands beyond the vertical direction into the horizontal direction, into the different cortical areas that I hinted at earlier in the introduction. And when we compare our data between different cortical areas from the posterior to the anterior, we notice that many neuronal subtypes, such as this one, would exhibit a preferential distribution in either the posterior or anterior side, but it forms a continuous gradient as it, let's say, gradually reduce when you walk from the posterior to anterior. So clusters like these can be found within every cortical layers. And here I'm showing you examples of like opposing pairs of neuronal subtypes between the anterior and posterior regions. So together, the neuronal subtype composition along this AP axis for the fetal cerebral cortex form a continuous spectrum, and that would align with what you imagine, say, if this patterning happens through the long-distance diffusion of morphogens that aligns with most other developmental features of the progenitor patterning. But this type of continuous difference does not explain why in the fetal cortex you also, why in the human cerebral cortex you also can observe the neighboring cortical areas forming a very sharp boundary between each other. So one of the well understood well-characterized example would be the boundary between the primary and secondary visual cortex, V1 and V2. So postnatally, they would even have a very dramatic lamination morphology difference that can be observed. But during the mid-gestation, again, the neurons are completely densely packed next to each other with no real differences to be seen morphologically. Between the area that will become the V1 and V2. So does that mean this specification between the two areas has not yet occurred? Our Murphy's data shows a dramatically different picture in that as early as the gestation week 20, the V1 and V2 are sharply delineated by this mutually exclusive distribution of neuronal subtypes that can be found across all cortical layers as well as the subplate. And we look at the distribution of these neuron subtypes across other cortical areas. We see that these green ones, green neurons found in the V2, they have some distribution and presence in other cortical areas as well, even though they are clearly higher in the V2. So aligning with that continuous difference that I described earlier. But the neurons in the V1, marked in purple and magenta here, they are truly unique. They are found only in the V1, but not anywhere else, like completely zero, suggesting that yeah, it may be the primary visual cortex has a very specialized specification program for these neuron subtypes. We also caught when exactly this border happens because 2 weeks ago at gestational week 18, we do not observe this sharp border, and the gene expression difference between V1 and V2 is not very dramatic. Well, we see it at week 20. Of course, we continue to see it at week 21, but also ever since then, in third trimester or even in adults, the V1 V2 neurons have very distinct transcriptomics differences. But these differences, like the gene that marks the difference between V1 and V2 during gestation week 20, does not fully overlap with those that can be marked in the third trimester or in the adult. So there are a lot of the temporary, dynamic, or say transient specification programs at play here. Another very interesting observation we have is that we are talking about this sharp border only found within the cortical plate, so the post-mitotic and post-migration neurons, but not in the migrating neurons or the progenitor cells. To make sure we didn't miss anything, because Murphy's is a method that targets a gene, targets a gene panel instead of the full transcriptome, we expand our analysis to the full transcriptome method of the Visium, which can measure all the genes but not at single cell resolution. Here we see the same conclusion where the boundary is only in the cortical plate and the subplate, but stopped at where the migrating neurons are. And if we look at the UMAP projection of these Visium spots, we see a dynamic trajectory where everything seems to share a origin and then split into two. So of course, underneath the subtype difference, many genes are differentially expressed between the V1 and V2, and many of them are more gradual, but with also noticeable examples such as the NPY exhibiting a almost binary switch-like behavior at the border. We can also expand our analysis to analyze genes that are outside our initial Murphy's gene panel by integrating it with single nucleus RNA sequencing obtained from consecutive tissue sections that we use for Murphy's. So in this way, we ensure that the two data sets have the identical cell composition in terms of the cell types and the ratio between different cell types. So this allows us to very accurately impute genes that are outside our Murphy's gene panel onto the Murphy's data to visualize its spatial pattern. As shown here, we were able to identify additional genes that show a very dramatic V1, V2 difference, and we were able to validate this imputation result with direct analysis. So everything I have talked about here so far with regard of the V1, V2 border is actually really, really surprising because such sharp border formation is not known to be in like most animal models, such as the mouse. So we look at the mouse data for different developmental stage, and we never see the genes that we use to see the human V1, V2 distinction to kind of show up the similar pattern in the mouse, or neither does it in the ferret. But when we expand our analysis to fetal macaque brain, we find that NPY actually marks the same V1, V2 border at almost the equivalent developmental, like mid-gestation developmental stage, suggesting that our finding is likely a primate-specific finding, though of course we want to confirm it by analyzing more different animal species. So we don't have a perfect explanation for why this border is happening. So this is one of the topics that I really hope to build more in my independent lab to have a mechanistic understanding. So right now we're kind of descriptive, but we can get some hints by doing some inferred bioinformatics analysis, such as gene ontology analysis, as well as receptor ligand based cell-cell communication analysis. And this is in collaboration with Chunhui and Liang at the BCH Research Computing. So we did identify that it seems that the layer 4 neurons specific to the V1 have a very early upregulation of synaptogenesis comparing to counterparts in other cortical areas. And we were able to validate this computational analysis with direct immunostaining for synaptic puncta. So in summary, we observed the two distinct modes of mid-gestation cortical arealization. One is continuous and aligns with the so-called proto-MIPE hypothesis that suggests cortical arealization happens due to morphogen gradient patterning the progenitors early on, building a continuous blueprint. But we also observe this discrete sharp border formation of cortical areas that favor the other hypothesis called the protocortex, in that it is the interaction of the cortex with external projections, such as those coming from different nucleus of the thalamus that shape the cortical area identity in a highly specific and localized manner. And both of these models have been proposed decades ago, but now we see that they are not mutually exclusive. They work together and may be of different dominant importance when it comes to different developmental stage or different areas. And with that, I like to summarize what we talked about so far, but mostly instead of reading from the summary here, I'd like to highlight a web-based interactive browser that we also built in collaboration with Chunhui and Liang here at Research Computing, where you can scan the QR code and you can explore the merfish data that we have in our study without bioinformatics background. So you can look at both like localization of cell types as well as genes of interest. So within the remaining time, I'll go really fast on a new direction that I'd like to pursue. And of course, now we established this very powerful pipeline of analyzing normal development. It's just natural that we want to expand it to the disease comparison. So here I'm looking at a disease brain with a very dramatic malformation called lissencephaly, where the cortical clarification is completely lost and the brain just looks completely smooth and the cortical layers is disrupted very dramatically. This has been known for decades with traditional histopathology characterization. But now with the help of merfish, we can really pinpoint that the shared neuronal subtypes found between control and disease, in the disease seem to have like most of the neuron types that you will find in the normal, but they all mislocalize in the cortical layer in a specific manner, that certain neurons go like up, certain neurons go down, that we can pinpoint all of these. And it gives us a hint on what is actually a cell type specific or developmental stage time understanding of the disease like pathogenesis process, which can be tested in like models such as organoids, which I don't have a lot of time to cover in today's talk. And we also see like certain neuronal subpopulation are exhibit a identity shift or just total loss. So yeah, with that, I like to have the acknowledgement slide for the Walsh Lab, where I perform most of the work I described in today's talk. And really, it's an amazing place with great colleagues and great colleagues such as Diane and many others. And I like to really thank Chris for his mentorship and his support as I am making the transition into my independent lab. So since starting the lab in October of last year, I'm very proud that we have built a very energetic team. And yeah, a lot of the work that today I'm only talking about the spatial transcriptomics aspect. We are that is very descriptive, right? Because it's all like snapshot of fixed tissue or frozen tissue. So you may wonder, how can we do anything mechanistically? And that's the other aspect of my lab that I will be pursuing. That's using the organoid models to study the development and diseases of the human cerebral cortex in a human-specific context. And leveraging direct analysis of the human tissue with spatial omics, we provide the benchmark to understand the strengths and limitations to make the most of it. Yeah, so with that, I'd like to thank you for your attention. And I don't know if we're going to go for questions right now or wait till Aakanksha's talk and have the questions together. Yeah, but thank you very much.
Okay, maybe we can ask, like, do we have any question for Dr. Xuyu? If not, we can go to the next talk, but you can think about the presentation which you actually presented. I think this is a really interesting talk. Yeah, if you have question, put that in the Q&A channel or raise your hand. We can unmute you. Okay, maybe we can go to Aakanksha. So Sham, you want to introduce
Yeah.
speaker?
Yeah, I'll introduce Aakanksha while she sits up. Hi everyone, I'm Shams from the Renthal Lab. It's my distinct pleasure today to introduce Dr. Aakanksha Jain. Aakanksha is no stranger to the Longwood community here, as she did her postdoctoral fellowship with Dr. Clifford Wolf at the Kirby Neurobiology Center in Children's Hospital, and many of us in the pain community have had the wonderful opportunity to work with her. Prior to her postdoc, she received her PhD at the Passaria Lab at UT Southwestern, and before that, she received her bachelor's from the National Institute of Technology in India. Currently, Aakanksha is an assistant professor at the Department of Immunology at the University of Washington. Her lab focuses on understanding the neuroimmune crosstalk underlying pain and inflammation. Her recent work has used transcriptomic-based approaches to reveal how the immune landscape changes with the generation and resolution of pain. This work has led to the interesting discovery of thrombospondin-1 as a suppressor of inflammatory pain, and her lab continues to identify mechanisms related to thrombospondin-1 and other receptor ligand interactions between immune cells and pain neurons to further our attempts to develop novel immunomodulatory therapeutics. So with that, please join me in welcoming Aakanksha at this seminar.
Thanks, Shams, and thanks, Liang and your team for this invitation. I already learned a lot in this webinar. As Shams mentioned, our lab, or my primary interest is in understanding how sensory neurons and immune cells interact with each other in the context of pain and inflammation. So just to orient everyone, we study the peripheral nervous system, which is where the pain is generated. Pain is triggered by specialized sensory neurons called nociceptors, and their cell body resides in the dorsal root ganglia, which sits right outside your spinal cord, outside the blood-brain barrier. And these nociceptors have long axons that innervate almost every tissue, including all the barrier sites where you can experience injury and feel pain, that leads generation of pain. And on the other hand, these cells also have primary axons going into the spinal cord, where the input then leads to the sensation, that the aversive feeling that we call pain. So the terminals here, if you just zoom into it, are very interesting bodies. They have of course, sodium and potassium channels that lead to action potential generation, but also transducer channels called the TRP channels that can transduce non-electrical signals like heat, pressure, vibration, etc., into an electrical signal of action potential. So TRPV1 is one of the most famous TRP channels that is the sensor for extreme heat, or the active ingredient in the chili pepper called capsaicin. And both of these will lead to the sensation of thermal pain, or the hot pain that you get with the chili pepper. And in most cases, pain is useful. This pain leads us to withdraw our hands when we touch a hot pan, etc. So we absolutely need this pain. In fact, people who do not have this protective pain due to some channelopathies have very limited lifespan because they keep hurting themselves, and as a result succumb to various injuries. So as I mentioned here, we need this acute protective pain because we want to withdraw from these potentially damaging stimulus. However, when there is an actual damage, for example, in case of postoperative pain or an ongoing immune response in autoimmune diseases, these cells can also directly see the immune activity in the tissues. So for example, immune cells make various ligands like prostaglandin, bradykinin, various cytokines like interleukin-1, and the receptors for these cytokines are also expressed on these nerve terminals. The downstream signaling leads to activation of various kinase cascades. One of the most important ones is this PKA, which is cyclic AMP-dependent protein kinase, which can directly phosphorylate some of these TRP channels, for example, TRPV1. This phosphorylation event leads to allosteric change in TRPV1 and leads to reduction in threshold. So for a channel to only respond to high temperatures like 52 degrees, now this starts responding at 37 degrees. So this leads to the feeling of tenderness or hypersensitivity, where something that was not painful before, like warm water, is now painful. And in most cases, this is also important. This is an adaptive pain because it leads us to guard that tissue as the recovery, the tissue wound healing progresses. But in the case of postoperative pain or autoimmune diseases, this is an unwanted outcome of inflammation. So one way to treat this kind of inflammatory pain is to either completely silence the nociceptors. Can you guys see my pointer?
Yeah, we
Okay.
can see that.
Okay. Oh, okay. So, so something like lidocaine, which would completely block the sodium channel, and you would not feel any pain. You will be completely numb. But in that case, the problem is that we also lose this protective pain. So when you go to the dentist, you get lidocaine, you're also at a risk of biting your tongue because you cannot feel anything. So the premise of my work is to find ways to preserve this protective mechanism while inhibiting this inflammatory pain. So to do. Okay. So one way to do that is to block the action of immune cells on these nociceptors, which will only inhibit this inflammatory cascade in these sensor neurons while preserving their normal danger detection system and bring this threshold back up to normal. So sounds easy, but it's not, because the immune cells in the tissues are very complex systems. Many of you might know an immune cells compartment consists of many types of cell types, many macrophages, dendritic cell types, as well as adaptive immune cells like T cells, B cells, etc., including resident cell types like Langerhans cells, mast cells within the skin. At the same time, sensory neuron compartment is also transcriptionally and functionally diverse. They can be defined by specific transcriptional markers, and these transcriptionally defined subsets are also responsible for specific function. For example, peptidergic neurons that can be defined by TRPV1 or CGRP expression are specifically designed to sense heat. Somatostatin neurons that are defined by SST expression are specifically designed to induce itch, and so on. So they're both very complex systems, transcriptionally and functionally. On top of all of that, when there is injury, the immune cell compartment undergo complete landscape shift. There are new cells that come in, like neutrophils and macrophages, and the existing cell types undergo dynamic transcriptional changes. So our idea was to find some sort of logic of interaction between these two compartments to maybe able to understand underlying principles of pain mechanisms that are mediated by immune cells. And what I've not mentioned yet is that activated neurons can also make neuropeptides that act on immune cells and regulate immune function. For the purpose of this talk and my work in Clifford's lab, I mostly focused on this direction, where we were looking for new immune ligands that change pain hypersensitivity and inflammatory pain. So to do this, we decided to map the receptor ligand interactome between the immune cells and sensor neurons. So before that, we just made a cell type agnostic receptor ligand interactome database. And we considered 3 types of interactions there. The number 1 type of interaction would be just a protein ligand and a protein receptor. So in this case, a protein ligand like IL-31, which is a cytokine made by immune cells, is a secreted ligand. It traditionally acts on the receptor, IL-31 receptor, which is expressed by sensory neurons. So this is your traditional receptor ligand interaction, which is mentioned by many of these cell type receptor ligand interactomes that you guys might have used. But in addition to this type of interactions, we also included other non-classical or traditional interactions per se. So another one is where the ligand is not actually a protein or a gene transcribed product. For example, prostaglandin E2 is a metabolite, and its production is controlled by an enzyme, prostaglandin synthase. The enzyme is transcriptionally controlled. within the immune cells. And then this metabolite is an intermediate product, which then acts on EP4 receptor that's expressed by sensory neurons. So this is not that traditional, and these kind of interactions are often missed in the cell-cell interactome databases that exist. And we, for the purpose of pain research, we also included this other type of interaction where the ligand does not directly interact with the receptor. For example, nerve growth factor that's made by macrophages and mast cells can alter TRPV1 expression or TRPV1 function, but ion channels like TRPV1 are not necessarily considered receptors in normal databases, but we included it in this setting. So to make this cell type interactome, I'm just gonna summarize a lot of the work. We used prior, We used prior databases that exist. So for example, Omnipath, which consists of many cell phone DB, cell chat DB that you guys might have used. These are high quality receptor ligand interactomes, but as I mentioned, we also were interested in finding new interactions or non-classical interactions. So for that, we used text mining systems to search through literature. These are PubMed's abstracts to identify specific languages that says something entity A binds to entity B. And on top of that, we also use structured databases that define what is an ion channel or what is a metabolite, etc. This was put through an assembling software, Indra. Sorry, I don't know why this is. Which was developed at Harvard Medical School in Peter Soger's lab. And as a result, we ended up with a receptor-ligand interactome. The output of Indra looks something like this. Because this is an evidence-based text mining software, every interaction that we call to be true comes associated with actual evidence from the literature. For example, IL-6 binding to IL-6 receptor, which is IL-6-ST. This interaction will be supported by actual language that's present in PubMed with their PubMed IDs. So if you don't like this interaction, if you think this is a questionable interaction, you can go to PubMed IDs and make sure if this is good or not. But we went through a lot of iterations to make sure that our database is largely correct. And this ended up with about 9,000 interactions that database that exists between that consist of all three types of interactions that I mentioned earlier. Sorry, I don't know why my slides are backwards. Okay, so this is our ongoing pipeline where we use prior databases and make an updatable cell-cell interactome. And I call it updatable because unlike cell phone DB or cell chat DB, which have static receptor ligand interactions, it's just an Excel file of existing interactions that they have already put together. Because our software uses PubMed, we can keep rerunning it and update our database to include newly reported interactions. So once we have this cell type agnostic receptor ligand interactome, we decided to use single cell sequencing to identify all the cell types that exist in the immune compartment and the neuronal compartment. And this will also give us the ligands and the receptors that are expressed by these individual compartments. Then the idea was to use some sort of statistical database or statistical software to identify interactions that exist in healthy or painful tissues, which will lead us to hypothesize new interactions and then validate. So this is a pipeline that we decided to go for. Sorry. Okay, so to look for the immune cell compartment changes, we use three types of pain models. They're all acute pain models in the skin. The first one is injection of zymogen. This is a TLR2 ligand coming from a yeast extract. Injection of this, just the powder from the yeast cell membrane, leads to an infectious anti-immune response that would normally be against bacteria or fungus. So this is our infectious associated pain model. The second one is a surgical incision, where we make an incision on the mouse paw and then suture it back up, which causes an invasive post-operative type of inflammation. And the third one is where we expose the mouse paw to UV light, which does not really break the UV barrier, the skin barrier, but causes some sort of inflammation. This is non-invasive, and it would be most equivalent to a sunburn, for example. And to confirm that there is actually pain hypersensitivity in all three models, we expose the injured mouse paw to a heat source, and we measure the amount of time it takes for the mouse to flinch. This is a standard model in the pain field. The less the amount of time it takes, the more hypersensitive the paw is. So here you can see, as in all three settings, there is reduction in the time it takes for the mouse to withdraw its paw, suggesting that there is hypersensitivity in all three models. But what stood out to us immediately is that in all three models, the kinetics of hypersensitivity was significantly different. For example, in Zymazan injection, which is an infectious model, there was hypersensitivity almost immediately after injection. However, in UV burn, it took these mice almost 2 days to develop significant hypersensitivity. So this already suggested that even though this is the same tissue and they're all three acute pain models, the mechanism that leads to pain in these models could be different. So to further look into it, we decided to do single cell sequencing to understand how the immune cell landscape is changing after these injuries. So for that, we isolated CD45 positive immune cells from all three models and the healthy contralateral skin. The immune cells were identified using FACS sorting, and we performed single cell sequencing. So this gave us an immune landscape from the healthy tissue. So these are cells that are expected to be in healthy skin. And upon injury, in painful tissue, there are all these new immune cell types coming. So we do know, now you can tell that there is a significant change undergoing within the immune compartment in these tissues. So when we first, so we decided to apply this to our cell-cell interactome, and we use CellChat-DB. And we did not use the database, but just the statistical part of CellChat-DB to identify significant receptor-ligand interactions. So just the mathematical modeling is what we relied on. And the output of cell-cell interactome looks like this. So we had all these immune cell types that we identified in single cell sequencing. And these were all the sensory neuron types. And I did not go into how this sensory neuron data was used from a previously published study from Clifford's lab. So when we combined this data set, we could identify interactions between individual immune cell types and sensory neuron types. And in this case, immune cell types were the sender of the signal, so we only considered the ligands that were made by these cell types, and the receiver were sensory neurons, so we only considered the receptors that were expressed by these different sensory neurons. And the darkness of each pixel is representative of the amount of the total number of significant interactions that we found. So you can see that there's a lot of interaction within macrophages, probably because macrophages compartment is undergoing a lot of change in these settings. And if we do that for all three immune settings, there were subtle differences between Zymazan, incision, and UV burn and their interactions that are taking place. For example, UV burn did not really have any of the granulocyte interactions. And within Zymazan, we found a lot of interactions with macrophages and the peptidergic neuron subset. And I want to point out that already it made sense that most of the interactions with these peptidergic neurons and NF3, which is A delta neurons, all these cell types are known to trigger pain, specifically heat hypersensitivity. All right, so if macrophages and neutrophils are the most interacting cell type with these sensory neurons, there are two ways they could be interacting. Either they are the most migratory cell type, so they are responsible for the most landscape shift within the sensory neurons, or they're undergoing injury-induced transcriptional programming change, and as a result, making new ligands that were not made before. So we looked at both of these types of changes. The number one is the amount of actual the cell infiltration for both neutrophils and macrophages. And for this, actually Liang's team with Aakanksha Jain helped us with this analysis, where he did this SC-CODA analysis, which does some sort of statistical analysis to understand if a specific cell type is undergoing significant change. And you can imagine, because the single cell subtypes, single cell experiments are only done in duplicates, or sometimes not even that, it can inflate these proportional changes. But this analysis allows us to do a mathematical modeling that gives us much more accurate statistical change within cell type proportions. So here it was very interesting, because you can see that both of these cell types are change in proportion at different time points. So for example, in zymozen, the macrophage and neutrophils are immediately infiltrating the tissue at the time. And this sad emoji is to remind you where we found the peak hypersensitivity in each of these settings. So you can see that the recruitment correlates with the time kinetics of pain hypersensitivity. It was at 4 hours for zymozen, 24 hours for incision, and for UV burn, both macrophage and neutrophils took almost 48 hours to reach their max, which is where we saw pain hypersensitivity, suggesting the recruitment of these cell types that we found by single cell sequencing is mirroring the pain hypersensitivity development. And second, we did differential gene expression analysis within every single cell type at all these different time points. These are a lot of numbers, but I'll just go through it one by one. So this is basically showing the actual number of differentially expressed genes. For example, 1,100 differentially expressed genes were found in CX3CR1 macrophages, which is a dermal macrophage subtype, at 4-hour time point in zymozen. And you can immediately see that the magnitude of transcriptional change also mirrors pain hypersensitivity. For example, at 4 hours in zymozen, macrophages are undergoing most significant change, and it starts resolving as the pain resolves. It takes longer in incision, and the peak reaches around 24 hours, while in UV burn, it takes almost 48 hours for these cell types to undergo any change. So this immediately suggests that the pain is actually associated with the changes in the immune compartment and is actually uncoupled from the injury itself, because the injury happened at 0-hour time point. So the neurons are not really responding to the injury, but responding to the changes in the immune compartment. So we also looked at individual genes that are being changed. So if you just basically focus on CX3CR1 high macrophages, which have the most transcriptional changes. And here, this is a dive-in plot actually was reported by Liang in this paper. So we took advantage of that. And it's a great way to visualize differential gene expression in if you have multiple different conditions. So in this case, we compared peak hypersensitivity for zymozen, incision, and UV burn, which is 4 hours, 24 hours, and 48 hours within CX3CR1 high macrophages. And you can see that there are a lot of the transcriptional changes are also very unique. There are changes in, for example, FGL2, which we only see in UV burn. And this makes sense because this is a gene associated with stromal compartment and keratinocytes and epithelial cells, which is what we think is undergoing damage after burn. On the other hand, genes like arginase 1, which is a wound healing gene, is changing in incision, which makes sense because this is an active wound. And finally, we found all these chemokines and inflammatory cytokines upregulated in zymozen, which are normally associated with antibacterial immunity. So this started giving us insights into differential pain mechanisms that might be playing a role in causing heat hypersensitivity in 3 different settings. So it already suggests that the pain treatments for different injuries could be much more personalized. Instead of giving everyone the same acetaminophen or same gabapentin opioids, we could use single-cell transcriptomics to identify specific gene signature specific to that injury to develop more precise medicine. And when we look at all of the peak genes that are changing across, so these are unique genes to all 3 injuries, but we also looked at commonly upregulated or downregulated genes. We found that a common signature where all of the NF-kappa B targets were upregulated at the time of injury, and a lot of the CREB targets and complement pathways were downregulated at the peak of hypersensitivity. So this suggests that if you want to find a common pain or analgesic, we could target NF-kappa B pathway or upregulate CREB back to normal. And we are currently following up on this in my lab. But I'll just tell you about how we go about to validate any of this data, because single cell transcriptomics at the end is just a pipeline to derive hypothesis and predict mechanisms, but not validate. So I'll tell you one of the story that we have followed up on. So if we focus on individual pixels, it consists of actual receptor ligand interactions. And this is the output from INTRA, where you can find the receptor ligand and the type of modality. For example, in this case, aldehyde dehydrogenase binding to interacting with MRGPRD1, but this interaction is not direct. Aldehyde dehydrogenase makes an intermediate beta-alanine, which then binds to MRGPRD. And this is the INTRA evidence. If you go to this PubMed ID, it will tell you that this interaction is actually real. And then finally, we also were able to identify which injury type this interaction is most associated with. So we were able to predict a lot of the known pathways. All the ones that are listed in black here are interactions that have already been associated with pain hypersensitivity, and people in the field are working on targeting these pathways. So the fact that we were able to already predict new pain or known pain pathways gave us a lot of confidence in our pipeline. But of course, because we did such a big study, we were able to identify a lot of new interactions, potential new pain mechanisms. And one of the ones we followed up on was THBS1 interacting with CD47. THBS1 is a protein, encodes for a protein ligand, thrombospondin 1, also called TSP1. And CD47 is the known receptor for this. And these are some of the PubMed IDs that can just give, this is a evidence for this interaction.
Sorry to interrupt, Aakanksha.
Yeah, this is maybe like 2 slides.
Yeah. Okay, great.
Okay, so just to tell you, when you have many of these interactions, how do we pick the top one to go after? The number one thing that we make sure is that the ligand is an induced ligand. It should not be present in the healthy settings because it should not be important for acute pain. And the second one is that the receptor should ideally be enriched in the pain neurons. So which is what happened here. The receptor CD47 is specifically expressed by pain neurons and not neurons that are responsible for gentle or normal touch, which always suggested that this interaction might be important for pain. So to validate this, we have done a lot of experiments, and I'm going to just have one slide to show this. But we were able to show using electrophysiology, calcioimaging, and animal behavior that TSP1 made by macrophages indeed talks to CD47 in sensory neurons to inhibit PKA, which I told you was a driver of pain hypersensitivity, and through TRPV1 modulation affects, downregulates or attenuates thermal hypersensitivity. So we were able to take the single cell sequencing data set and predict a novel interaction, and were able to validate this interaction to identify new mechanisms for pain. And now we are going into neuropathic pain and chronic pain to identify new chronic pain regulators. So with that, I would like to thank everyone in the Wolf Lab, especially my students who work on this project, Will Renthal and Shams, who is here, who helped us with some of the data analysis. And a big thanks to the bioinformatics core who helped me with this paper. All the data is made available online. This is my current lab, and I should just plug in, we are hiring postdocs and students if you are interested in this type of project.
Oh, hi, I see a question from Monica. Let me see how I can unmute her.
Yeah, I can do that for you. I think now you can talk, Monica, now.
Yeah, hi. Thank you so much, Aakanksha. That was a great talk. I wanted to ask a specific question. So I think this study is really interesting. And another reason why I thought that this kind of analysis is important is because drugs like gabapentin, they work for some, but they don't work for some, even with the same condition. Like, for example, like an L5S1 disc compression, which causes numbness. So if the situation is like this, how do you plan to take samples from these patients?
Yeah, so pain is a very complicated problem. There are many different problems. One, we don't even know how to quantify pain in these patients. And then, of course, not every drug works for every patient. So I'm working with patients with phantom limb who have phantom limb pain. I don't know if you've heard of it, but this is pain people experience after amputation, which is similar to a nerve injury pain that you just mentioned because of crush. And in this, some people experience pain, some people don't experience pain. my current hypothesis, or the favorite hypothesis, actually immune compartment also defines if you are going to be susceptible to develop chronic pain or not. And of course, and then also define if you are likely able to respond to a specific treatment or not. And I'm saying that because we're already finding that based on asymptomatic or symptomatic pain, the immune compartment is quite different in patients with at least phantom limb. And these people are also have kind of found that in diabetic neuropathy. So of course, my favorite hypothesis is that first you should look into the immune compartment, both circulating and tissue resident compartment, but it's a complex problem.
Got it. Thank you.
Well, I have a data science question for the two panelists here. I noticed that there is actually a really strong importance of temporal changes, with Xuyu noticing strong change between gestational weeks 18 and 20 in the cortex, and Aakanksha's noticing time-dependent effects of pain. How do you guys think about the 4D, the temporal space in the context of single-cell omics in your work?
Yeah, that's a great question. It's especially from the developmental time point of view, we're always thinking about that. So the omics analysis is currently kind of by nature snapshots, right? So the temporal trajectory is something that we kind of need to infer by connecting the data points together. But of course, now we see analytical methods getting better and better. Like previously, we had the like pseudo time analysis, and now there can be AI tools that can be used. There are lots that we can do there. But then, of course, if we can combine that with a mechanistic modeling such as the organoid to really see things unfold, it will strengthen it even further.
Yeah, I mean, I agree. The one thing I would like to say is that a lot of these things they are predictive. At the end of this is all modeling. So very important to go back and to find whatever you are specifically interested in. Like you did a lot of the validation and we do a lot of validation. It's important to do functional validation at different time points.
Of course, the finer we can do the smaller the gap between different time points, the finer we can reconstruct the trajectory. Because what if it takes a weird turn somewhere in the middle? So catching the very transient programs are very exciting in a way. Like for instance, I talked about the V1, V2 border at week 20. That was a transient program, like the genes that were upregulated in the V1 at that time is not something that you can reconstruct by like connecting an earlier time points to the adult and predict, okay, these are the genes that go through a linear time course. So yeah, to be able to catch them is key, but also requires a bit of luck in terms of what's available. Like now it's going to be tougher for the developmental tissues. As the NIH is not going to fund the fetal tissue research for a while.
Wonderful. Well, I think we're running out of time. Should we conclude here?
Or do we have any other question from the audience? I actually maybe I can ask one question here if there's no more question from the audience. I think I would like to ask this question for our postdoc. I'm not sure how many postdocs here right now. Well, this question is for both Xuyu and Aakanksha. So as former postdoc at BCH who successfully moved into a faculty position, could you share any experiences or advice that might be helpful for our researchers here at the BCH?
Okay, maybe I'll start first. Aakanksha, I don't know if you went on the job market for the 25 cycle or you started like before and you interviewed the cycle before?
Yeah, I interviewed in 24.
Okay, yeah. So I interviewed during the spring of 25, so the most recent cycle. So I think a lot of people got the impression that last year was a really tough year. But I think that of course later in the spring we hear about all the news about the interviews being canceled or even rumors of offers being withdrawn or position being closed. But to be fair, when I sent out the application in the fall of 24, it was like it was like a ton of opening. So even if a lot of them got canceled, last year was not like that bad. So then I think a lot of the things that we talk about, like how pessimistic about things are going to be, are just being concerned about the worst of the worst scenario, and which may not be the case, and doing the best that you can regardless of the external circumstances is the key. This year is not going to be too bad. A lot of new. I still have my nature jobs and Cell Korea like notifications of all the new positions that I see. Some of those are the comeback of the canceled positions last year. Some of those are new ones. So in total, it's also I think there's a dynamic balance there. So yeah, probably this is not really about strategy to succeed, but I'm just saying, it's there's no need to be despair. There's still a job market, and I think it's still relatively strong.
Yeah, I just want to say that I think of course they go in waves, but just try to make a research program that's different from your current boss, and is unique. And then apply to places where you would have a nice supportive environment and people are actually excited about what you're working on. Yeah. So I think just propose something that's exciting. And I think there's always a place for that everywhere.
And I know people at Children's people are doing extremely competitive, right? Because when I was rehearsing my chalk talk in the whiteboard and places, every conference room that I go into the CLS, the whiteboard is filled by someone else's chalk talk. So I think around this time of the year, it's where it happens. You're going to notice that too. So yeah, there's no shortage of people we can learn from. Yeah, but yeah, I think one thing in particular would be good if postdocs can get any experience on what a chalk talk is like before going on the interview. Like in my case, I've never sat at the chalk talk. It's always kept as such a cryptic activity that it's not open to the outside. And I don't know if there's a ways to engage people, at least to come in as audience. Maybe they don't ask questions, but then sit at the back to see what it's like.
Great. Thank you so much for sharing this kind of valuable experiences. I think that's really helpful for our researchers here who want to look for the faculty position. Do we have any other question from the audience? We run out of time a little bit. Sorry about that. Okay, if there's no more question, well, thank you so much for two speakers, Xuyu and Aakanksha, and also the co-host here to introduce our speaker and monitor the Q&A session. And thanks everyone for joining this meeting. And you all have a great day.
Okay, thank you.
Thank you.